Thanks everyone for joining what is our second last Spatial Conversations webinar for the year. I'm really excited that we've had so much interest from people in the series so far, and I'm excited to see this talk, particularly around the use of TMAs, which I get a lot of questions about. So for those who are new, I'm Brett, the Market Development Manager for ANZ for Spatial, and my colleague from MS.
So my name is Marta Gabrielska. I'm an account manager at Millennium Science, responsible for 10x Genomics. We are a distributor of many suppliers, but 10x Genomics is my favorite. So I'm really happy to host you here as well. And maybe I will just jump to introduce our speaker, actually two speakers. So as you can see, we are upgrading today. We are giving you two speakers instead of one. So today we have Professor David Gallego Ortega from University of Technology, Sydney. He's an acting head of School of Biomedical Engineering, head of Functional Genomics Laboratory, lots of heads, and director of Single Cell Technology Facility. And with him, we have Dr. Alan Faes. I probably pronounced it incorrectly, forgive me. He's a molecular biologist and geneticist working with David, and he's leading the Respiratory Bioinformatics and Molecular Biology Lab, also at University of Technology, Sydney. So just before we jump and start, if you have any questions, please put them in the chat. We will ask them and encourage everyone for a discussion after the presentation. Also, you will be allowed also to ask them by raising your hand and we will unmute you and you can ask the questions as well. So I think with that, we can start talking about the exciting topic of mapping the immunosuppressive landscape of the premetastatic niche in breast cancer. To you, David and Alan.
Thank you very much. Thank you, Marta, and thank you for 10x and Millennium for the invitation for these Spatial Conversations. I think this is very exciting for us to be presenting this project that we've been working together. So just a little bit of housekeeping. So I will be presenting the main kind of directions of the project and where are we coming from and the data that generated the motivation to use the Xenium platform and the way we did it. So I think this is important for this talk. And then Alan is going to go and explain a lot more in detail about the Xenium data and how he's analyzing the data in combination with the single cell RNA-seq that we had already. So here we go, see how we go. Okay, so of course, this talk is about a kind of cell that is called myeloid-derived suppressor cells. But this comes, as you all know, that the tumor microenvironment is quite a relevant compartment for the initiation and the progression of cancer, and of course in breast cancer as well, and is critical for the development of metastatic disease. So these myeloid-derived suppressor cells are immature myeloid cells. They are two types of them. They are the granulocytic type and the monocytic type. They resemble those canonical markers of neutrophils and monocytes, but they are also quite immature, so they are not fully differentiated. And the main thing is that they contribute in many ways for cancer progression and metastatic dissemination. So they they're able to induce EMT and favor the invasion of the cancer cells through the stroma. They, of course, do immune suppression. That's the role of them, where they target cytotoxic T cells on natural killer cells and prevent them to reject the tumor. They are ECM remodelers. They also promote angiogenesis, protect the cells that are the cancer cells that are in circulation, so the circulating tumor cells, and also help them to home into the premetastatic niche by promoting systemic inflammation in the secondary organs. In this case, today we're going to be talking about the lung as the secondary organ for metastatic dissemination. So previously, we did a lot of work into looking using single cell RNA-seq to map what was the mechanism that activated and attracted those myeloid-derived suppressor cells to the tumor in the first place, coming from the bone marrow. They release, they have an early release in response to the cytokines released by the cancer cells, but also from the tumor microenvironment. So using single-cell RNA-seq back in the day, we were able to look into where are the cell types within the tumor microenvironment that they were contributing for that attraction and education of myeloid-derived suppressor cells. But to better study this mechanism, and also because we became very interested in metastatic breast cancer and how these cells are able to prepare that pre-metastatic niche, we designed a different kind of mouse model, and we used the 67NR and 41.2 cell line, which is injectable. So we do introductory injections of these cell lines. They resemble triple-negative breast cancer. And as you can see, the 67NR is not metastatic, while the 41.2, they're highly metastatic. So using this pair of cells, what we did was to inject into the mice, and then we'll see that we use time points to follow the progression of the disease, not only in the tumors, but also in the lungs. So on the left-hand side at the bottom, you can see that the level of metastatic dissemination is completely, is black and white. And also we use flow cytometry to look into what was the distribution of myeloid-derived suppressor cells using canonical markers, which can also result in picking up neutrophils in those lungs. And as you can see there, and also across the time points, so we have from 0 to 4 weeks during the development of the metastatic disease. As you can see in the 41 in the green bars on the right-hand side, you see that there is a massive accumulation in the lungs using flow cytometry. And then we also did sorting and confirmed that these cells look like myeloid-derived suppressor cells. They are granulocytic in nature. So as I said before, we designed 2 time points. The week 2, which is we looking at the distribution and the infiltration of those myeloid cells into the lungs. We didn't see any cancer cells at that stage, even in the metastatic one. So we deem this state as pre-metastatic. And then we have the metastatic one, which is week 4, as I showed you before, full of metastasis and full of inflammation of increased number of myeloid-derived suppressor cells. And as I said before, we basically follow these 2 models, 2 organs, 2 time points. And the analysis that we did was based on single-cell RNA-seq using the 10x Genomics Chromium platform. This is not fixation, so this was before the Flex came into place. And then just recently, we decided to do also a Xenium analysis in the platform. So this is data from the single-cell RNA-seq that we got, and as you see, we profiled the tumors and the lungs, but very briefly, but we took the messages that we could see those neutrophils. I'm not sure if you see my arrow. I hope you do. The neutrophils in the lungs coming very, very highly represented and more abundant. So with that, basically, we extracted those neutrophils from those lungs, and then we characterized the subclonal resolution, I guess. We basically subclustering those neutrophils to see what were the different transcriptional profiles in there. And then we identified, again, very quickly, a unique species of these neutrophils or granulocytes that they were consistently represented in the metastatic situation, but they were not present in the samples that did not have metastasis. So we call these ones prometastatic, and this is a little bit of the definition that we did. All the clusters at the top are neutrophils, and they're consistent with canonical neutrophils and differentiated in different stages of activation. And the ones, cluster 1, 2, 3 at the bottom of the dotted line, we classified as neutrophils. This was also based on their ability to suppress the immune system. So they were immune suppressive populations. As you can see, the green one is actually very, very immune suppressive compared to any of the other populations. And I'll hand over to Alan to tell you about the Xenium data.
Okay, so now we're sort of going to go through the Xenium. So the for this sort of experiment, to try and do this across multiple slides, that would cost a fortune, a small fortune. So basically what was done, slides, sections were punched out, and basically this tissue array was created. Now, this was then run on the Xenium platform with the tissue atlas, which involves 379 genes plus 100 custom genes as well, which really overlapped with the neutrophil genes that we're quite interested in this project. Now, this work was done by Laura and Emily here, and I got involved with the analysis side. Something that was really important, the first thing was checking the gene quality. And we can see that the pre-designed genes were quite highly expressed, and also our custom genes were expressed as well, really well as well, at least higher than the negative controls, which means they were expressed. We also can look at transcripts per cell, and you can see the transcripts are sort of over the board of these circles here, indicating that there wasn't any problems with the scan of the Xenium. And then we can also look at clustering, and I'll go through that in a little bit more detail in a moment. So what I did was I took the data first from Xenium Explorer and downloaded it so we could actually run it through Seurat. We use an in-house script, and if anyone's interested in that, we're happy to share. Now, filtered out poor quality transcripts based on the definition of that 10x uses in their Xenium Explorer. And we dropped the number of minimum features to 1. That might seem quite ridiculous when we talk about single cell data, but for this Xenium data, we want to make sure we retain every single cell that actually has some expression, so we don't lose or have gaps in our profiling. So if we just quickly look at some of the QC metrics, we can see transcript counts were pretty even across the samples. Cell size didn't really change too much, but cell number did, and we'll see in a moment based on the slides, and it makes sense. We had a lot more cells in the tumors that really are just really packed in there, while the lungs are much more dispersed. You've got much more open areas, and that's where the oxygen exchange occurs. Now, identifying cell types is a little bit more difficult than with single cell because you don't have all the genes that you'd normally have. So we only had 479. So it's good to have an atlas here. So we actually, my lab has developed this mouse lung atlas, which we use predominantly to identify the markers, specific markers for each cell type. And this is basically a UMAP that was generated from the data, and you can see we've got close to 1 million cells, so you need a relatively good computer to run this, but it's also important to realize that the clusters really overlap one another. They're not as unique as you'd normally see in single cell data, and it's just because you don't have all the same transcripts you normally would. So using our atlas, we're able to identify the different cell types. We've got cancer, airway epithelium, really nicely separates immune cells, and epithelium and stroma. And then once we identify these, we can really nicely just upload them very directly into the Xenium Explorer, and this works really seamlessly. So this is what we did, and we can see we've got the tissues above and below. What we observe here in yellow, I've highlighted the cancer cells. In blue, we've got the inflammatory cells. Purple really nicely highlights the airway epithelial cells, and in pink, we've got the structural cells, so they're the alveolar epithelial cells. And you can see that much nicer over here. Now, if we zoom in on this, this is how it looks just with the normal staining. If we overlay the expression, one of the biggest things we can see is that the cancer cells here in yellow, there is actually a few cancer cells that are away from the main tumor. And you'd lose this resolution if you were to not stain or not have this Xenium data. So it worked really well. And the other thing that we can see, the segmentation of the cells or this package works really well. I thought I'd have to go in and trace out a lot of cells. This was all done automatically. So it was quite a nice experience for us. So how did this look when we looked over the all the samples? Well, if we look in our metastatic cancer mice, we can see here in yellow, the tumors are 100% tumor cells, which makes sense. And then here we can see at week 4, when we're expecting to see metastatic tumors, we can see that there's a nice tumor here, and you can see a really nice margin where you can see that. And then here we can see these little microtumors also present. And then obviously the tumor from week 4 is just as explicit. Now, what we observed here in the non-metastatic, in the lung, we see basically no tumors at 2 weeks like we saw above, but here we also see no tumors here at 4 weeks as well. And this is what we would expect. Now, one thing to keep in mind, if we took this sample and ran it through traditional 10x, so this was before Flex, we actually wouldn't actually see any cancer cells. And this is because of the processing, and these cells seem to lyse and not actually be picked up with 10x. This problem has sort of been solved with Flex, but this is important to realize when you're looking at slightly older data. So we then wanted to identify neutrophils. This is where the atlases are perfect. So you look at one of these atlases, we get a neutrophil signature. This is where our neutrophils sit. And we can see this signature is quite strong in neutrophils and unique. Now we can take this back to our UMAP. And basically, we highlighted that 24 and 7 sort of hold our neutrophils, or a subset, a part of 7. We did have to do some reclustering there. Now, what we do is we identify which cells are neutrophils, and you can see that here in red. And then, really nicely, you can overlay the expression profile signatures. So if I zoom in here, we can see that our neutrophil classifications really nicely overlap with our neutrophil signatures based on the atlas. And really, it's just a nice form of verification that we're actually identifying neutrophils. Now, how did this look with disease? Well, what we found was in the metastatic mice that the neutrophils here in white are really defined to the non-cancerous regions, and they don't penetrate the cancer very well. And while we observe in the lung tissue of the non-metastatic, we don't see many or any neutrophils. If we sort of quantify this, we can see there's a very significant increase in neutrophils at week 4 and barely any here. One thing we do see, we do actually get a little bit of penetration into the tumors, but you can see that these percentages are much lower. So, we then wanted to see, can we identify the original subtypes? And what you can do is we basically made a subcluster, use the signatures from the original single cell, and identify these exact cell types. We can then put this back into the Xenium data, and you can see basically these are colored green, red, and yellow. They also not really sitting in the tumors, on the outside of the tumors, but it gives you some positional information about these neutrophils. And we can then run additional analyses to see, are they more likely closer to the tumor than other types of neutrophils? And it's also important that the non-metastatic has little to no. So in conclusion, the metastatic for T1/2 has increased abundance of neutrophils, and compared to non-metastatic cancer, and the Xenium shows neutrophils are mainly not penetrating the tumor, and the Xenium allows for retention of cancer cells that you may have lost using other single cell techniques. So I'll just leave it here for David for the acknowledgements.
Well, basically, I'd like to acknowledge all the people involved in this project. So this is only the tip of the iceberg of the project. It's much bigger, but I just highlighted the people from the 3 different labs that are involved. So my lab, Favaldes Moras, and Alan's lab, working together in this project. And I also wanted to acknowledge that the Xenium machine that we use is part of a grant that it was funded with UTS, and the Garvan Institute was led by the Garvan, but UTS was part of it, funded by the Cancer Institute New South Wales, and it sits actually at the Garvan Institute. And this is the platform that we use for this Xenium run. And of course, all the other institutions involved and the funders that supported this work. And thank you very much for your attention.
Thank you, David and Alan. That was really interesting. Just a reminder to everyone in our audience, feel free to put your question in the chat or raise your hand, because it's spatial conversations. So that's the main part, is the conversation bit. So maybe while you are warming up to ask your questions, I bet Brett has a question.
Always, Marta, always. Yeah, thanks very much for the talk. I think it's a really great example of how even a small data set can be, or a small number of slides can generate really interesting data. I do get asked a lot about TMAs because they are, obviously, as you pointed out, a cost-effective approach. Sometimes we see that people, when they put TMAs onto slides, they don't get data back from all of the sections, and handling TMAs can be challenging. Can I ask how you found working with TMAs on Xenium from a more practical side?
Yeah, no, look, we basically have in our design, since we have a mouse model, so we have 2 cores per block. So we have 2 different regions of each of the tissues. And also we have 3 animals, or 3 replicates of the same one. So we have 6 samples in reality that are talking about the same thing. That is to accommodate for any potential loss of tissue in the slide. Now, in our experience, and I also like to acknowledge this was done by the histology department at the Garvan, that they are fantastic handling these samples. We didn't have any issue, any detachment, any problem running with the Xenium, any other than the problem that happens with normal histology. So of course, if the quality of the tissue is not there, it is not there for anything. But we didn't see, we ran, of course, a number of sections before and after the Xenium, just to check quality before we run into the big experiment. And we didn't see any difference. It was very, very simple and very easy. All the process is absolutely smooth. Now, one of the things that I think that is also very important is that we were able to do H&E in the slide that we run in Xenium. And I think this is important because you also keep that extra information. We did run the segmentation kit from 10x to do that segmentation that Alan showed, that is actually automatically picking really well those cells. But I think it's important because now we were able to have those 2 images back to back, just the immunohistochemistry and that. So you can recover that after the analysis.
So we have already 2 questions in the chat. First question from Andrew Lucas. So he's interested, it would be ambitious to look earlier than 2 weeks to maybe see the preconditioning interactions. What do you think?
Yes, of course. So we do have in the data set in the Xenium DME, we do have healthy lung. So we can compare so with the animals that did not have any tumors, and then we can compare from 0, if you want, or no disease, to a early disease, which is 2 weeks. The question is that we are very focused on the inflammation. So we really need to have a little bit of kick from the tumor to develop those connections. Early, yes, the earlier the better, because if we want to propose early detection methods or markers, then that's the key. And from our point of view, inflammation happens before metastasis. So the earlier you can identify that specific signatures of inflammation, the better for the prediction of metastasis. Absolutely.
Andrew is nodding. He texted, he's nodding. We have another question from Eamon Colan. I hope I pronounced it correctly. What were the advantages of re-uploading to the Xenium Explorer rather than doing the spatial analysis with Surat? And was the upload of annotations to Xenium Explorer, sorry, it just popped, just via a CSV file?
Yeah, so reason to re-upload. So most of the analysis will be done using Surat, but to visualize the Xenium Explorer was fantastic. I was using a very, let's say, mediocre laptop, and I was able to visualize with the Explorer quite readily. Also, the Explorer allows you to do all your overlays, exact expression, and you can also save it and then send it to other people as well that may not have coding experience, and they can just open that in Xenium Explorer and then visualize it themselves. So using the Xenium Explorer as a final point where you make your figures, I think is fantastic. The brunt of your analysis, your differential expression and everything like that, I would still be using Surat. The upload, simple CSV file. Again, you just have to have your cell names, and then the whatever your definition is next to it. It's pretty simple.
If I was doing analysis now, it would be like I would be really tempted to, if I can put cell names, I would give them like crazy names. Like I don't know. I would give maybe some political names for the cancer cells. Anyway, sorry about that. I have another question from Erin Cole. Did you select the cores for each conditions as regions of interest on the Explorer and export the cell IDs this way, or export all and annotate with condition in Surat after?
Yeah, so yeah, at this stage, you can't just select the region and then download the matrix from it. So I took the first image, downloaded all the data, then used the regional analysis to identify the cell types, and then added that as a meta column into the definitions into a meta column in the Surat object after. There isn't an easy way to do it, but there is some nice scripts that, again, we've developed, which we're happy to share to aid in that process.
Cool. And I actually have a technical question. So when you were showing the clusters that you had from initial Xenium, like the first cluster with the red cancer cells, you like clustered cancer cells as one cluster, while for me it looks like a bit two separate clusters. Were there differences between these two parts?
Yeah. So that was very level one, trying to just identify the structural cells. And then I don't know if you saw the UMAP later on. We had like 28 clusters. And there you can really disentangle. But one thing it's important to realize, the neutrophils sort of merge into the structural cells, and the structural cells merge into. The only thing that was really separate was the airway epithelium, which was this nice small cluster up the top. So it's depending, when you get 5,000 genes, it's probably not going to be a problem. Things will be nicely. But when you're only doing 500 genes, you just need to be prepared that everything's going to sort of merge into each other. You probably need to do a bit of reclustering.
And if I can add, I think this is the value of having the two data sets. So we can interchange information accordingly, and then we can top up the weaknesses from each of the platforms. So again, if we did the 5,000, it wasn't available when we did it. So if we did the 5,000, probably it will be a lot more resolution in there. But since we have the single cell data created from the same animals in the other with the other technology, then we can basically expand, and we just need the link. What are the markers in one data set that links to the other one? And so then we can do that.
I think you did like really amazing with what was available at that time. I'm especially referring to what you achieved in terms of neutrophils, because I know what I've learned working in this role is neutrophils are very tricky to work with. And now with GEMX technology, it's actually easier, and with Flex as well. But like you did it even before, so like it's really, really impressive, and also shows that Xenium can help out with that as well.
Yeah, absolutely. It was I mean, obviously neutrophils is one of the main focus of my lab. So we've been working with neutrophils for a long
You
period.
have gold hands for neutrophils then.
And well, you know, our protocols are, I would say, they're really well optimized to make sure that we pick them up and we don't lose them. We've been doing a lot of flow cytometry as well, so we have a very clear idea of when we lose them and when we don't, and how can we enrich them.
You might get some people trying to get in touch with you about that.
Exactly.
Yeah,
I'm putting a
absolutely.
note, neutrophil expert. Yeah.
I'm just going to ask last question from me. I think we're no more from the audience. Last question for me would be, obviously you did a 100 gene custom panel. I do ask this question a lot, but I was just interested in hearing from you how you found the experience and the performance of the custom panel versus the off-the-shelf genes.
Look, that was our first quality control that we did, because our 100 custom panel comes from the key genes that we identified in the single cell data that we have created. So we were expecting to find them and spatially locate them in the Xenium now, because this basically builds our story. So those 100 genes, we wanted them to be robust and represented. And in the quality control, I think that was one of the slides, we could see there's no difference. So they do perform the same as the off-the-shelf atlas. Now, I have to also say that we got a lot of support from at the design moment and the design level from 10x. So that was a very good experience. So Laura did all that kind of analysis and to see, make sure that I don't know exactly what the algorithms are to identify where the Prof seed and all that. They're very well defined. We had no issues in any of the genes of the 100 that we use. So very, very happy with that. Yeah. Great.
Just to add to that, having an atlas or something to map against to make sure that you sanity check that the genes are actually in the right locations is always important as well. And from what we can see, they're just as good as the main panel. They're in the right positions, in the right cell types. There hasn't been any unexpected aberrant expression or potential off-specific binding that we've been able to observe.
Excellent. Fantastic talk, guys. I will quickly say to everyone, I did put in the chat a new tool from the Broad Institute, which they have just launched in the last few days to assist with cell annotations, leveraging the CZI database and tens of thousands of data from single cell experiments. So for those people here, I think that might be of interest. It's brand new off the shelf. It was a collaboration with 10x Genomics, and we're excited to see that come to people. But with that, I think we're out of questions. We're out of time, but I'd really, if you could fill out your feedback, we really appreciate it. We're preparing for next year and what people would like to see. And I would like to thank David and Alain for a fantastic short talk.
Thank you. Thanks for the invitation.
Thank you, guys.